Hi, this is Miss Litton, and this is a very small 7th period. Today, say hi.
Hi.
Okay, so I found this today, and I thought it was really, really helpful, and I got it from Khan Academy, which is a great place to get some information to review. So take a look at this picture. The pink part up here, glycolysis, who does, I put it on Instagram. If you want to sit on it. Okay, who does glycolysis? All cells. Good. And then you have a choice. Remember when we get to pyruvic acid, it's either you have oxygen or you don't have oxygen, right? So if you have oxygen, you get to go down the aerobic pathway, and you're going to move into what organelle? Mitochondria. If you don't have any oxygen, then you're going to stay in the cytoplasm. Stay in the cytoplasm, and you will do fermentation. Remember, our big ticket item here is we're trying to oxidize or break down glucose, right? And temporarily, we give the electrons, the hydrogens to who? Who do we give them to temporarily? They don't know. Do you know? What?
The electron transfer.
Before that, yes, we are.
Reduce. No.
Yes, we reduce something. What do we reduce?
The
The NAD, right? Remember when we reduce some NAD and make some ATP too. So when you're reducing that NAD, you're reducing it with the electrons / hydrogens from glucose, and you're transferring it, yes?
Yes.
Okay, so you just do that in a bunch of little small steps. Ultimately, when you're all done, then you're going to, if you have oxygen, you're then going to go to the top of the electron transport chain, and you're going to take all the NAD and FAD that you reduced, and what are you going to do to that NAD and FAD? You're going to what it?
Spend it.
How do you spend it? It's reduced NAD. What are we going to do? Oxidize it. We're going to oxidize it. And they're going to drop off the electrons at the top of the electron transport chain. That's when you do your what? Right? That's when you do your reductase, cytochrome oxidase, and you take hydrogen ions from the matrix and put it out into the intermembrane space. So when it comes back in, do you remember what enzyme protein it comes back in through? ATP
Synthase complex.
Yes, ATP synthase complex, that's when you make some ATP. Okay, so that's like the big picture. I have another, I'm going to go off of our review just for a minute. I think I do have it. Where is it? Not here. Not here. Not here. Don't say it. Okay, here. All right. So this is a review I had made last year, and I just kind of remembered that I had it. So cellular respiration, the whole purpose, oxidize glucose in order to generate what? ATP. Do you want me to print this and put it in the Google Classroom stream? Yes. Okay, I will. Okay, so you're transferring those electrons and hydrogens that are in the glucose to NAD and FAD, right? And then you're ultimately going to take those to the ETC. We say that's like taking it to the bank, and you cash it out for some what?
Energy.
Yeah, what kind of energy? ATP. Are you guys thinking you don't know the right answers because you're not saying them? They don't care about us. They don't care. All right. So if I look at the steps, glycolysis, transition prep, Krebs citric acid cycle, ETC, did I just show you the steps of aerobic respiration or anaerobic respiration? Aerobic respiration. Okay. So these are my very crappy, diagrams that I drew, but I think you will understand them. Let's take a look at all the different kind of steps where they occur. This is supposed to represent the cytoplasm. This is supposed to represent a what? Mitochondria. Good. What's the centermost part called? Matrix. What's this part right here called? Intermembrane space. Say it again. Intermembrane space. All right. So when we'll break down all the steps, the first step, glycolysis, 10 different steps, 10 different enzymes. What do we get to walk away with when we're done with glycolysis? Two what? ATP and two what? Reduced NAD. NAD. Okay? So here, once you do glycolysis, you walk away with 2 ATP and 2 reduced NAD. I need to preface, when you have reduced NAD as a product, what did you have as a reactant? Oxidized NAD. That's important. Put a pin in that. We'll come back to that with fermentation. Okay, following glycolysis, as long as we have plenty of oxygen, what are we going to do?
Aerobic respiration.
Aerobic respiration. And the next step is called what? Transition prep. And we call it transition, we're transitioning into the mitochondria, and prep, we're getting ready for the what? Krebs cycle. Okay, so here we're going to move in, and we're going to take our 2 pyruvic acids. We're going to do transition prep. When we're done with transition prep, do you understand how I look at this diagram right here? Talk with your bio buddy. What did I show right here? What am I modeling right here for you? What takes place? Oldest bio buddy, tell the youngest bio buddy. Go. Hopefully you know. They send me money. How do you send somebody money?
You have like stickers out, and you buy watching, and then you can send them, and like you get a percentage of it.
They sent me a panda? Thank you, person who sent me a panda. I don't know what that means. Is the panda in the zoo? Oh.
It's good.
Oh, does that mean sideways? Okay.
Yeah, that's you.
That's me sideways. We are TikToking live. Is that okay? Okay. All right. All right. Thank you for the panda.
They send you like.
Anyway, so basically what's
Oh, thank you for my panda. All right. I don't understand it, but I like pandas. All right. So let's look right here. Transition prep, and we go to. Remember when we did it? We go. What's the who? Two carbon dioxides. Good. And we make 2 acetyl-CoA. Put 1 away. Why do we put 1 away?
So you can do the
Krebs cycle twice.
Right, okay. So this represents the 2 CO2. There's your 2 reduced NAD, and we have 2 acetyl-CoA. So then the next step, and I think this is going to get messy, but you're going to like it, right? Of course. Okay, so take a look at what I did here on the citric acid cycle, Krebs cycle. How many times do we do it? Twice. And I will PDF this for you so you can have it on the Google Classroom. Okay, so if we look right here, look at the things that are coming off of it. I have an ATP coming off of that. Now, I'm going to review that again with you, but that kind of formation of ATP is called substrate level phosphorylation. Remember that? That's just the handoff, right? Okay, and it says 3 reduced NAD. Do you remember those 3 NAD when we make them? 2 plus 4 is 6. What? Do the bit, get 5. Do the bit, get 4. Make ATP, reduce an FAD, reduce an NAD. Perfect. So those are your 3. And you're going to do it 2 times, so 2 times 3 is 6. That right there shows you how important the Krebs citric acid cycle is in generating ATP ultimately. Because when you do that one, you get 6 NAD, whereas glycolysis, we only got what? 2. And in transition prep, we only got 2. Okay, so now we have 6 just from this one. And we have something else to take to the bank. Every time we do the Krebs citric acid cycle, what? FAD. Those are not as powerful as those strong NAD, right? Because the strong NAD are going to get us how many ATP? 3. Okay, and the FAD will only get us 2. Perfect. Okay, do you see the 2 CO2 coming off here?
Yeah.
Okay, but since we do it twice, how many CO2s come off? 4. And in transition prep, we had 2 more, so there's what? 6. So that's a total of 6 carbon dioxides, and so that means we had 6 carbons. How many carbons were in glucose? 6. So we just did the banking for all of that, right? We took care of all of them. All right, and then what comes after the Krebs citric acid cycle?
The
Electron transport chain. Okay, let me see. That picture is going to get worse. So beautiful, so beautiful. Okay, so we're going to take all of our NADs, see our NADs from glycolysis, our NADs from transition prep, our NADs and FADs from the Krebs citric acid cycle, and we're going to take them to the top of the ETC. Does everybody get to go to the tippy top?
No.
Who gets to go to the tippy top? And by top, we mean top energetically, who has the most energy, right? Okay, and they're going to drop off their electrons. They're going to na-na-na-na-na-na-na, right? And what do we mean by na-na-na-na? We have a series of redox reactions where the electrons are dropped off, passed to the next, passed to the next, passed to the next. But intermittently along here at what are the 3 places? Reductase, cytochrome, oxidase. At those 3 places, you're going to take hydrogen ions from the matrix, the center part of the mitochondria, and you're going to put it out in intermembrane space. Okay? Out on the in the intermembrane space, are there any questions or anything I have to answer? Okay.
Someone just said DNA replication is hard.
Do you want me to do a TikTok about DNA replication? I will do that. We're not quite there yet, but I will. Okay. Okay, so we concentrate the hydrogens out in the inner membrane space. Remember, when we concentrate them out there in that space, there's a difference in concentration. What else? A difference in pH and a difference in charge. Good. And so now they really want to move from the inner membrane space into the what? Yes. Yes. Where do they want to move? From the inner membrane space back into the? No, there is no outer membrane space. Back into the matrix, okay? And when they move back into, see the little arrow right here? Let me, sparkle pen, because it's so pretty. So pretty. Okay, so when those hydrogens come back in, that's when you make some ATP, right? Remember right here, that's that ATP synthase what? Complex. Perfect. Okay, I think that, oh yes, and I have some more pictures. That was with oxygen. If we don't have any oxygen, the only thing we can do is the first step, which is called what? Glycolysis. We do the first step. And then the second step without any oxygen has to be what? Fermentation. And when you do the whole reason you do fermentation, the entire reason you do fermentation is because when you do glycolysis, you reduce some NAD and make some ATP. When you reduce that NAD, pretty soon all your NAD starts to build up. Why doesn't it build up when we do aerobic respiration?
Because it's going to be taken to the Krebs cycle.
No, it's not because it's taken to the Krebs cycle, to the electron transport chain, and we drop the electrons off. That's when those NADs and FADs get oxidized so you can reuse them again. But if you don't have an electron transport chain, and why don't we have an electron transport chain to use in anaerobic respiration? No oxygen, so the chain will back up. Chain won't work, okay? So we can't drop off any more electrons. The electron, all the receptors are full for all the electrons. So now we need another way to oxidize that NAD. That's why you do fermentation. So here in fermentation, you still do glycolysis in the cytoplasm. You're still in the cytoplasm. You can't go into the mitochondria because you don't have any oxygen. Take your 2 pyruvic acids. There are 2 choices, but for us, there's only one choice. What's the one choice we can do?
Lactic acid.
We can form lactic acid. And lactic acid, when it builds up in our muscles, makes our muscles sore. Okay, plants and yeast, they do a different strategy. They'll take the pyruvic acid and they will reduce it down into alcohol. This alcohol has how many carbons in it? 2. So you lose carbon as CO2. Okay, in a nutshell. All right? Okay, I got to remember to print that off for you. Okay. All right. Other thing I wanted to review with you are the types of phosphorylation. Now, to phosphorylate means to add on a phosphate group onto something, to phosphorylate. Could be anything that you're adding it onto. What we're talking about right now is adding a phosphate group onto ADP to make it what? ATP. Okay? But you can phosphorylate other things. You can phosphorylate enzymes, in the cell cycle, etc. But substrate level phosphorylation, that's the handoff. That's like what we do in glycolysis, where we just take the phosphate group and with an enzyme we transfer it to a different molecule. Oxidative phosphorylation. What do you hear in oxidative phosphorylation? That's what you do in your electron transport chain because you need oxygen at the bottom of the chain to accept the electrons. If you want, I can PDF this too or take a picture of it if you want that. Okay. So oxygen at the bottom of the electron transport chain, and then it forms what? Water. Okay. We haven't learned the next one yet in this class. In Honors Bio, we haven't learned this, but if you need this, AP Bio has already reviewed and taught photosynthesis, and there are videos in there. But photophosphorylation, we could deduce from this that photo must require not oxygen, but what? Light. Okay? And so, in that case, in photophosphorylation, that's the next thing we're going to learn on Monday, is using light to phosphorylate. Okay? So I wanted to review those 3. Let's do Minecraft glycolysis. Okay. Now, is this the truth right here? No, because really it makes how many ATP? 4. But to get the party started, we had to spend 2, and then we get 4, so we net out 2, and we reduce 2 NAD. When we reduce those NAD, where do they get the electrons from to reduce that NAD? From glucose, and that happens the whole rest of the way. We're just repeating that process over and over again. All right. Mitochondria structure, we've already reviewed that, but you want to be comfortable with that. Let's talk about transition prep. Okay? So, it's so annoying every time. Okay, I won't fix it right now. Transition prep. Okay, now, just ignore that over there. So on transition prep, we have 2 pyruvic acids, and because we have oxygen, we can take these 2 pyruvic acids, and we're going to convert them by removing a what from each one? Carbon. We're going to remove a carbon. And when we remove carbon in cellular respiration, what does it come off as? Carbon dioxide. Okay, so we remove it as carbon dioxide. And then, so that's when we do the little song, we go, so we remember that, right? And we reduce some what? NAD. How many NAD do we reduce? 2. Okay, and that's what we do, 2-acetyl-CoA, put 1 away. The next step, do you have any big questions on that one? Do you need me to follow through or show you this carbons? Are you good? I think we've hit it pretty hard. In class, we learned this. Okay, let's do Krebs citric acid cycle. So we take acetyl-CoA, we bind a 2 carbon with a 4 carbon, and we make a 6 carbon. Then what we say as do the bit, that's nothing biological, that's just a made-up litinism. When we do the bit, we reduce NAD and we pull off a carbon dioxide. Okay, and then by pulling off that carbon, we're down to C5. We do the bit again, and we're down to C4. And this C4 is the same C4 that we started with just less energy, because we're going to take a phosphate away from it so we can make an ATP. We're going to take away some electrons so we can reduce an FAD, take away some electrons so we reduce an NAD. And how many times are we going to do that? Twice. Okay, perfect. All right, electron transport chain time. Is this hard? Is this part hard? No. Right? Okay, so NAD and FAD, let me make this a little bigger. Okay, NAD and FAD drop off their electrons. NAD at the top, FAD partway down. What's the significance of starting partway down?
Lower energy. It generates less ATP eventually.
Yeah, generates less ATP because you miss the first reductase. So you don't put out as many hydrogens into the inner membrane space. Is there anything I need to be aware of? Okay. You don't put out as many hydrogens into the inner membrane space. But the NAD drops off those electrons. We nana, right? Passing those electrons along. And at 3 places, reductase, cytochrome, oxidase, we're putting hydrogens into the inner membrane space. That's great. The more of those we accrue, the more want to come back in. And when they come back in, it's like turning a turnstile. We will make some what? ATP. Good. All right. At the bottom of the chain, who's the final electron acceptor? Oxygen forming water. Good. All right. I think that's it on that. Before we sing, let's go over fermentation. Fermentation is a strategy. Darn it. Okay. Remember, the sole point of fermentation is to do what? Oxidize what? NAD. Oxidize your NAD. Oxidize your NAD. So you have 2 different ways to go about it. Us, only one way. You do glycolysis, which you can see right here, reduce some NAD and make some ATP. Oh no, we don't have any oxygen. What are we going to do? Fermentation. So you and I will take the pyruvic acid, which is a 3-carbon molecule, and it's still a 3-carbon molecule, but the bonds are rearranged. And we actually reduce the pyruvate into lactic acid, which makes our muscles sore. But in the process of reducing the pyruvic acid, whenever you reduce something, you also what? Oxidize something. And so who are we going to oxidize? The NAD that we reduced in glycolysis. So we can use it over again. Plants and yeast do a different strategy. They convert it to ethanol. Okay, and they will release the CO2 as a result of that. Last thing, where's the pizza? Maybe the pizza. Oh, here's the pizza. It's a terrible picture. But I want to remind you, in everything that I've discussed today, we used glucose as our starting point, right? But we could use proteins, and we could also use what? Fats. They all feed in different places. Remember, fats are made of glycerol and 3 long what? Fatty acid chains. And if it's an animal fat, what kind of fat is it? Saturated fat. And we can take those saturated fats, those chains, and snip them into groups of 2, and those could be acetyl-CoAs. All right? And protein's the same. Catabolism, is that breaking or building?
Breaking.
And anabolism is building. Okay? Ready to sing?
Yes.
Okay. All right, here we go. This little song, it's posted on TikTok if you want to learn it, or on my YouTube. Yes, thank you. Okay? Ready? What's the whole thing called? Cellular respiration. What's the first step?
Glycolysis.
Who does it? All cells. Where do they do it? In the cytoplasm. What do you start out with? Glucose. How many carbons?
6.
How many? 6. What are we going to do?
Break it in half.
How many steps?
10.
How many enzymes?
10.
What are these called?
2 pyruvic acids.
And you
Reduce some NAD and make some ATP. 2.
Oh, we have plenty of oxygen. What should we do?
Aerobic respiration.
What's the next step called? Transition prep. Here we go. And you
Reduce some NAD and 2 acetyl-CoA. Put 1 away.
What's the next step called?
Krebs cycle.
Another name?
Citric acid cycle.
Krebs citric acid. Good. How many times are we going to do it?
2.
Why are we going to do it twice?
2 acetyl-CoAs.
Good. And every time we do the bit, we are what?
Reducing NAD and
Pulling off a carbon.
dioxide.
Okay, here we go. 2 plus 4 is 6. Do the bit, get 5. Do the bit, get 4. Make an ATP. Reduce an FAD. Reduce an NAD. And 2 plus 4 is 6. Do the bit, get 5. Do the bit, get 4. Make an ATP. Reduce an FAD. Reduce an NAD. And stop. Gather all your reduced NADs and FADs and take them to the top of the ETC. Na-na-na-na-na-na-na. Reductase, reductase, cytochrome oxidase. And you make some ATP. A lot of it, a lot of it, a lot of it, or a lot of it, a lot of it. Who catches electrons at the bottom?
Oxygen.
Forming
Water.
Pick up your glucose. What are we going to do?
Break it in half.
Steps.
10.
Enzymes
10.
called
2 pyruvic acid.
and you.
Reduce some NAD and make some ATP. 2.
Oh no, we don't have any oxygen. What are we going to do?
Fermentation.
How many choices do we have?
2.
You and I make lactic acid. Makes our muscles sore. Plants and yeast make ethanol. Either way. Alcohol. Sorry, it's really alcohol. Sorry. Alcohol. Either way, you oxidize your NAD. Yay! Oh yeah, ethanol, alcohol. It's all good. All right. Any other questions? Are we good? Are we done? Okay. What? Oh, want to do some Hello Smarts? Okay, let's do some Hello Smarts. Let me think where I have the most. I think here. Let's log in as something fun we're going to do this weekend. Should I still live stream this? I guess I can show them the questions. We're going to ask some questions. Actually, oh my gosh, I didn't even think of this. You can jump in right now and do these questions with us.
No way.
Yes, they can. Yeah. Okay, so let me tell you what you need to do if you want to try some of our practice questions. Is this safe? All right. Hopefully. You want to go on to Hello Smart. Oh, yeah, here. Put this in front. There. Can you see it?
Wait,
Anybody watching it? Can they see it?
Did I have anything on there that I didn't want? No, I'm fine. I think so.
Okay, so you go to hellosmart.com. Oh, this is a great way for people to practice. Go to hellosmart.com. You want to join my class code, which is 878464. I'm going to remove the ones that are in here right now. Maybe. Okay. All right. And let's just go in as a nickname that's appropriate.
T-Bone.
You want me to remove Dr. Phil's brand?
No.
Oh, okay. Okay, so you can go to HelloSmart. Oh, sorry, there you are. HelloSmart.com. And then just put in the code 878464. 878464. 4, and then you can answer questions with us in real time. That's cool.
Okay, someone's TikTok baddie. Maybe they're from TikTok.
Maybe.
That was my last one.
Okay. He did not. Where is he?
I don't know. He told me he was
Hashtag disappointment.
Upset because we braided his hair in English.
All right. I can't remember if this has the big questions or not, but let's give it a go. Oh, this one is short. It only has 4 questions. Okay, and for those of you on YouTube, I'm going to show you the questions right now. So if you're at home and you're doing this, commit these answers to a piece of paper. Yeah, that's if they're live streaming, they could do it. But if they're watching this at home, like for our regular YouTube channel, yeah. So there's question 2. Here's question 3. A lot of it. And here's question 4. We might have 2 people out there. Do we have 2 people out there? Because there's like 14 of us here after school today. Unless somebody logged in twice. I don't know. Welcome. Welcome to our class. Okay. Okay, here we go. Sorry, Bob. I'm sorry. I'm sorry, Bob. I'm so sorry. I just have to keep going because we have to go home. We're tired. Okay, let's talk about it. The greatest contributor, peeps, here. The greatest contributor of electrons to the electron transport chain is?
Citric acid.
Citric acid cycle. Okay, why? Because it has 6 NADs, yeah? Number 2, approximately how many ATP are generated for each NAD? 3. Yay! You got 100% on that. Oxygen becomes water.
Oh, it was me, my bad.
Oh, man.
I hit the wrong one.
Yeah, 10. Okay, I think there's some more questions here. No way, way. A lot of it, or a lot of it. Okay, here you go.
Even though I know it might be wrong. And then my brother finally has proof that I don't know everything, so I'm hoping that he doesn't find it.
Okay, at home, I'm going to show you the questions so you can practice. Okay, answer your questions, you guys. Yeah. Oh, I'm sure you're doing a great job. I'm already proud of you. There's 3. There's question 2. Here's question 1 if you're at home. Code is 878464. Oh.
Okay.
Wait, what's the code?
878464. 878464 at hellosmart.com. Okay, are we close? You need more time? Okay, at TikTok land, map means multiple answers possible.
I don't know, like reduce the things.
Go back to that one.
That one thing where you didn't know that it was that. What do you mean by try?
Okay, are we ready? Who needs more time? Okay, let's end it. All right. So where does glycolysis occur? Cytoplasm and all cells, right? Okay, stop for just a minute. 2 of you, maybe you're in TikTok land, I don't know, hashtag no shame, but not the mitochondria, because remember, glycolysis occurs in the cytoplasm. Good. And it doesn't occur on any membranes. All right. Question 2. Oh my. Okay. Let's talk about this one. Okay. Okay. Back here. Let's talk about this one. Okay. Which of the following are needed for glycolysis to occur? Do we need any oxygen? No. Not for glycolysis. The only time you need oxygen is if you're going to move into the mitochondria, transition prep, Krebs cycle. You really needed an electron transport chain. Do we need glucose? Yes. NAD-ox? Yes, because when we reduced some NAD, it had to be what first? Oxidized. NAD-ox. Do we need reduced NAD? No, that's not a reactant. That would be considered a product. Chlorophyll is for photosynthesis. Did anybody pick that? Okay, good, no. Mitochondria. Do we need any mitochondria yet? No. We could be a prokaryotic cell. We don't need any mitochondria for that. Do we need ATP? Yes. How many? 2 to get the party started. And then we'll get 4 ATP out and net 2. All right. Do we need any pyruvic acid? Nope. No. Why? That is a what? Product. Okay. And then number 3. Okay. Which of the following are products? Not oxygen. Anybody put that? Yes. Somebody did. No. T-bone. T-bone. Okay. Glucose is not a product. It would be a what? Reactant. And NAD-ox would be a reactant. Reduce some NAD product. Chlorophyll? No. Different reaction? No. And ATP? Yes. And pyruvic acid? Yes. Okay. Hey, I think that's it. Oh, here. Yeah, here's one more. Here you go. Let's try to get 100 on all of them.
So far, I have to actually.
Okay.
Jordan, we're so proud of you.
You gotta move your thing. I'm gonna kill your crop one of these days.
What's fermentation? Oh my god.
Okay, at home I'm showing you the questions, so do these at home. There's only 2. Which of these is not true of fermentation?
Oh, not true.
Yes, not true. And which are the possible products of fermentation? You got this. I believe in you.
There's more than one possible
Oh, shoot. Map.
Oh my gosh, can we shout someone out?
What?
Emma V. Paul said this is so helpful actually.
Yay, Emma. I'm glad you find this helpful. Yay.
Yay, Emma, we love you.
Oh, that's nice. See?
This thing is live, and so she'll
What?
see this comment and like maybe.
We're very nice.
I like that.
Okay, do we need more time? Emma, you need more time, Emma? All right, here we go. Okay, come back to me. Guys, I know it's Friday and we want to go home, but let's finish. Which of these is not true of fermentation? Do you gain only 2 ATP? Yes. You walk away with 2. Does it occur in the cytoplasm? Yes. NADH donates electrons to a transport chain? No. We're not in the mitochondria. It does begin with glucose and yeast to it. And oh, Lord in heaven. Which are possible products of fermentation? Lactic acid makes our muscles sore. Glucose would be a reactant. Alcohol? Yes. Carbon dioxide? Yes. Oxygen, acetyl-CoA. All right, that's good. The end. Yay! Hope this was helpful to you. Have a great rest of your day, weekend. Make good choices.
Okay, how do I end this? Oh, the X button. Okay, bye.
How much coin did we make from that live stream?
32 people.
There's no coin.
Oh, 32 people watched it?
Yeah.
That's like an entire.
I don't even know.
That's
32
an entire
No, that's.
class right there.
It makes up for the people that didn't show up.
I don't even know 32 people.
374.
So you got 5.
374 viewers.
Oh!
I got a gift from Lexi. I got 5.
What does that mean?
I think that's like how much is it? I think when you put your like PayPal or something to it, you can like get money. I don't know. And like over time.
374 viewers? That's a lot, right?
It makes up for Richard. I know.
What is 5 vibe?
I think
Someone
that's
sent you a panda.
But
That's
did she
like
give
99 cents right
Wait, no,
there.
didn't she give her a panda?
Yeah, someone gave her 99 cents.
You gotta shout her out.
What is gift points versus contributors?
I'm not sure. It's like one person
Contributor
gave you
who
5
gave it to
points.
you.
I don't have enough
Oh,
followers to know that.
okay.
what do you even do with that? Like, is
I
it
think
like,
you cash them out, but like maybe you probably have to
What
have
do
a
you mean?
minimum.
yeah.
Okay, manage my account. Balance.
5 gift points.
0.
What? No, because that's like how many coins you have. 'cause you have
Oh.
points.
Oh yeah.
We go on.
Unless you've like spent money on TikTok already.
Yeah.
No.
How do you
Wait,
spend
normally
money on?
can't
Yeah,
you,
like
see the
apparently
people
there's
who
like this
contributed?
coin thing. So when you pay for coins, then you can give other
There should be
people
like a cash out
coins.
section.
I don't know.
All right.
Oh, this was my saying about it.
Oh, didn't somebody want a what is
DNA
it?
replication.
DNA replication.
Yeah.
DNA replication is
And
hard.
mitosis, there's one.
Yeah.
If you want to know what people want, look at this thing. I did it because I thought, oh, I want to be helpful. Okay, little
And
did
people
I
spam
know. 315 comments of what they wanted.
Oh. We just did the Calvin and Krebs cycle.
Yeah.
Or
Cellular
Krebs.
respiration, enzymes, osmosis, diffuse. These are, I said, is there anything you want me to go over? Yeah. Okay, that's a
Oh
lot.
wait, this thing.
Yes.
You know the thing that's like, it's like the glucose molecule plus like 6O2 is going to. Is that cellular respiration
yes.
or is that?
This
the
right
That's
here.
not
respiration
glycolysis,
today.
right?
And guess what?
This is
They
the
come
whole
only
thing
by
of cellular
one
respiration.
color of
Okay.
paint.
Yeah.
What's wrong with that? Is it purple?
No, it's
Oh.
blue.
I like purple.
But yeah, now I can't paint the board because there was only one color.
You can just paint it.
Oh, sorry, I forgot to turn this off. My bad. I hope you're having a good weekend.